Good morning everyone. Today we discuss about the topic is gram staining. First one is what is gram staining? Gram staining is a differential bacterial staining. It's one of the technique. It's used to differentiate bacteria into differentiate the gram positive and gram negative types according to their cell wall composition. Next, history of gram staining. The technique was introduced in 1884 by Danish bacteriologist Hans Christian Gram. He is first identified by the 1852 to September 13 to 1938, November 14. He developed this staining technique to identify bacteria causing pneumonia. Later it became a popular method to classify bacteria into gram positive and gram negative types. These are the differential types of staining protocols and procedure. First one is simple staining, differential staining and specialized staining. Gram staining is a differential staining method. It used to identify and differentiate bacteria into two groups such as gram positive and gram negative bacteria. This classification is based on the physical properties of the cell wall of bacteria. First one, principle of gram staining. Gram staining and differentiation are based on the cell wall structure and composition of bacteria. So bacteria having cell walls with a thick layer of peptidoglycan will resist decolorization of primary stain and appear violet or purple color. Bacteria have a thin peptidoglycan layer with lesser cross-linkage loss primary stain during decolorizing and gain counter stain appearing pink or red. So in aqueous solution of crystal violet dye, their molecules dissociate into crystal violet plus ions and Cl minus ions. These ions easily penetrate the cell wall components of both positive and negative bacteria. The Cv plus ions interact with negatively charged components of the cell wall. The Gram's iodine is added as moderate strain. The iodine interacts with the Cv ion and forms Cv1 complex within the cytoplasm and cell membrane and cell wall layers. When decolorizing solution, ethanol or mixture of ethanol and acetone is added to interact with the lipids in their cell wall. The outer membrane of the gram-negative bacterial cell wall is dissolved, exposing the peptidoglycan layer. The peptidoglycan layer is thin, less cross-linking in the gram-negative cell wall, hence becoming leaky. This causes cell to lose most of CVI complexes. So these decolorizing solution dehydrates the peptidoglycan layer, trapping all CVI complexes inside the cell wall, and the bacteria retain the purple or violet color of crystal violet. So this image represents the gram positive bacteria cell wall structure and the gram negative bacteria cell wall structures. Next one, four types of reagents are used for the gram staining method. First one, crystal violet, it also known as the primary stain. Next one is iodine. Iodine is otherwise called as modern stain because it increases the affinity of dyes or a stain for smear. Third one, alcohol or ethanol. It's referred as decolorizing agent that remove the stain from the specimen. Next one, safranin. It's referred as a counter stain. Next one, procedure of the gram staining. First is prepare a smear of the bacteria culture and heat fix in the smear. Next, added the few drops of crystal violet and imparts the purple color to all cells. Wash the slide with distilled water for few seconds. Next, the smear is added with iodine solution. The slide was then washed with the distilled water or tap water. Next, added the ethyl alcohol and take rest for the few minutes and washed with the tap water. The stain lost purple color and appears colorless. The bacteria that retain crystal violet and appears purples are classified as gram positive bacteria, while that no retain stain and lost the color after decolorization are classified as gram negative bacteria. These are the flow chart for gram staining procedure. Since the gram-positive bacteria already retain purple color, they are not affected by counter stain, but gram-negative bacteria are colorless, so they directly stained by the safranin. Thus, gram-positive appear purple and gram-negative appear red or pink color. The gram-positive bacteria cell walls are thick and chemically simple. So, in contrast, gram-negative bacteria cell walls are thin and complex multi-layer structure. When treated with alcohol gram-positive bacteria, are not purple, the CVI complex and cells remain the purples. The gram-negative bacteria have lipopolysaccharide layer. Then absence in gram-positive bacteria to which CVI complex is leaked and resulting in decolorization with later take the counter stain and appears red. So application of gram staining used in research to classify the bacteria into gram-positive and gram-negative, used in diagnostic lab for identification of pathogens, and the hospital for using spectrum and antibiotic treatment before completed identification of bacteria, used to study the morphology of bacteria or microorganisms. That's all about the gram staining. Thank you everyone.